Hey all, welcome to Share Trek. This is Raj here, and friends, I welcome you to the 5th episode of the Genomics for Investor series. Today we are going to delve into the fascinating world of CRISPR-Cas9. It's a buzzword and many people are talking about it for the last so many years. And as a genomics investor, you need to understand what CRISPR-Cas9 is, how it works, and what are the use of CRISPR-Cas9 and what are the various variations that have been developed since then. A groundbreaking genetic technology like CRISPR-Cas9 allows us to edit DNA with precision. Don't worry if this sounds complex. I'm here to break it down into simple, easy to understand concepts for everyone, regardless of your background or education level. So let's talk about an introduction to CRISPR-Cas9. It's the bacterial defense system, believe it or not. CRISPR-Cas9 is like the superhero of the genetic world, and it all began with bacteria. These tiny organisms use CRISPR-Cas9 as a defense mechanism against their arch nemesis, the viruses or phages. Step 1 is creating the CRISPR array, which is basically the bacterial memory. Imagine bacteria having a little memory bank in their DNA called a CRISPR array. This memory bank contains short sequence of genetic code from past viral infections, kind of like postcards from their battles with viruses. Or it could be a Rolodex where each entry is that of a virus. It's the identity of a virus. So a CRISPR sequence typically consists of a series of short repeating DNA sequences interspersed with unique sequences derived from past viral infections or other foreign DNA. These unique sequences are what provide the system with the ability to recognize and target specific DNA sequences for editing. The area of bacterial DNA that contains series of short repeating DNA sequences interspersed with unique sequences derived from past viral infection is called the CRISPR array. The reason why it's called as CRISPR array is because it is repeating sequences. It's a palindromic sequence which is repeated again and again. A palindrome is a set of alphabets which reads the same whether you read it from right to left or from left to right. So it's a mirror image of itself, so that whenever you read it from right to left, you'll get the same sequence as if you're reading it from left to right. So that's why it is a short palindromic sequence. Now, if you look at Cas proteins are the molecular Caesar, is the other part of the CRISPR-Cas9 equation. Alongside the CRISPR array, bacteria produce Cas proteins, with one of them being Cas9. Cas9 is like a pair of molecular Caesars. In this superhero story, later on, we'll see many other Cas proteins that play the role of molecular Caesars with varying levels of efficiency and precision. Now, let us look at this whole sequence from the beginning. Let us imagine a viral invasion happens, the viral villain arrives. When a virus invades a bacteria, it's like the villain showing up. The bacterium recognizes the virus and takes a small piece of its genetic material called the protospacer and adds it to its CRISPR array. This is akin to saving a piece of the villain's costume after having defeated the virus. Then we look at the CRISPR RNA. The bacterium then writes a little handbook called CRISPR RNA or CRRNA based on the stored viral sequence. This handbook carries all the information needed to recognize the viral invader if it ever returns again. Just like we know the mRNA is a copy of a gene, the same way CRRNA is a copy of the viral sequence. It's also referred to as protospacer stored in the CRISPR array. So if you visualize reimagine the CRISPR array again, you have a repeated sequence of nucleotides, which are palindromes, and in between each of these repeated sequence of palindromes is the CRRNA or protospacer or the piece of DNA taken from the invading virus and stored there for future use. And then we look at the showdown when a virus comes back again. So if the same virus comes back again, Cas9 teams up with CRRNA, which is a copy of the virus DNA, which is stored in the CRISPR section and thrown out, just like a gene throws out mRNA. CRISPR section has got the viral signature of the virus that has reinvaded the bacteria. It throws out a copy which is called a CRRNA, and CRRNA teams up with Cas9 and it goes to the virus, and the CRRNA has the same pattern in the virus and it locks onto it, and the Cas9 just cuts the virus at that spot. So the virus is now broken, and the virus will try to repair itself using the repair mechanisms of the bacteria, but the repair won't be perfect and the virus is destroyed. This is how the whole thing happens. And the DNA repair is never successful, and the virus is compromised and it no longer can damage the bacteria. So this is how the whole system works. Now, the scientists can use the same sequence in the lab to target a particular part of the DNA and edit it. But that is a topic for the next episode or some subsequent episodes. But right now you have understood what CRISPR is and what Cas9 is and how the two of them work to protect a bacteria from an invading virus. Now I'm going to walk you through this with a video sequence so that you can actually visualize it. So always a revision of the same thing is really advantageous to understand it much more better. So the way I would like to explain this to you is that let us imagine a virus invades a bacterial cell, and then when the invasion happens, the virus deposits its DNA into the bacteria. There are different types of virus. Some viruses are RNA viruses, but CRISPR-Cas9 is basically focused on DNA viruses. So the DNA that is deposited by the virus into the bacterium has got a signature, and that signature is thought to be the spacer or protospacer. The bacteria takes that protospacer from the virus after defeating it and integrates it into the CRISPR area. Out here you are seeing the CRISPR area where you have got a green square, which is the latest protospacer taken from the latest invading virus. And then you have a pink square, a lavender square, an orange square, and a blue square. Those squares are respectively signatures of various past viruses that had invaded this bacteria and was subdued by the bacteria. So whenever this bacteria multiplies, it's going to pass this on to its offspring, and the offspring will collect more. That way the entire bacteria colony is capable of preserving itself and defending itself against future viruses of the same type. This process is called adaptation. Next, let us assume that there are molecular seizures that are present within the bacteria because we already know that they exist. These are Cas proteins. So Cas9 is a molecular seizure, and when the same virus invades the bacteria again, what happens is that a copy of the protospacer is thrown out, and that is the CRRNA or the CRISPR RNA. And the CRISPR RNA contains the DNA sequence at one end, which is the green portion that you see here. And what happens with this is that it teams up with the CRISPR and then goes on to the entire viral DNA sequence of the repeat entry of the virus. And the green section of the CRRNA finds its own match in the virus's signature, and then it latches onto the DNA in that portion. And when it latches, the Cas9 chops off the viral DNA and cuts it into two pieces. And now the viral DNA tries to repair itself using the bacteria's repair mechanism, but it cannot. So the viral DNA is compromised, and therefore it's not able to harm the bacteria. So this is the whole sequence. So friends, I think we should always take small bite-sized approach at any given time so that we can understand the concept and commit it to memory and our understanding thoroughly, and then go to the next step. So sometime down the line in one of our next or future episodes, I'll be talking about how the scientists have used this defense mechanism of the bacteria in order to enable us to edit DNA almost in most of the places that we want. So with that said, I think I would like to bring this segment to an end. We like to keep it short, we like to keep it simple, and this is vital education for a genomics investor. We'll also be examining base editing and prime editing going on forward. We'll also be talking about the disadvantages and advantages of CRISPR-Cas9, the transfer mechanism. We're going to talk about terms like autologous and allogenic. We're also going to talk about concepts like in vivo and ex vivo. So stay tuned. And if you're not yet subscribed, please subscribe for more information like this, the knowledgeable videos out here. And also, if you're already a subscriber, please consider becoming a member so that you can support this channel. Thanks and have a great day. Bye for now.